Hello, everyone. Welcome to today's MASTS double webinar. So we'll have two speakers today who are Dr. Alex Thomson, a molecular phycologist based at SAMS, and Ximena Vega, a PhD student based at the University of Stirling. So a couple of housekeeping notes to begin with. After both presentations today, we will have the live Q&A session. So if you'd like to ask a question, please use the Q&A box that you'll see at the bottom of your screen. And questions can be submitted at any point. So if you have a question for Alex and you don't want to forget it, you can type that out. And then we'll work through them after both presentations. This session is scheduled to end around 1:50 p.m. UK time, but we may go over due to questions. But don't worry, because the entire webinar will be recorded. So if you miss something, you can't stay for the entire session, or you want to share it with one of your colleagues who wasn't able to join, you'll be able to find the recording on the MAST YouTube channel, along with all of our other previous webinars as well. So a quick introduction to our speakers. Alex works on molecular techniques to ecological and applied questions. Their current work focuses on the ecology and physiology of Antarctic snow algae. Ximena's research focuses on the dynamics between glaciers and the ocean and the role these systems play in the carbon cycle in polar regions through biooptics and remote sensing. So that's enough from me. I'd firstly like to hand over to Alex today.
Hi, good afternoon, and thanks very much to Bethan for that introduction, and also for inviting me to the MASTS seminar series. I'm Alex. I work at SAMS in Oban as part of Matt Davies' group, and we focus on a small group of organisms that are quite interesting bunch of photosynthetic algae known as snow and ice algae. And in particular, we're interested in the biology of these algae and how that determines their occurrence and distribution in the Antarctic. Last year, I was lucky enough to be part of the MASTS Saltire exchange program, and that resulted in a couple of collaborations with two groups, Chris Hewlett and Alex Ditton from Nord University in Norway, in Bodo, and Thomas Leyer from the Fraunhofer Institute in Potsdam, who runs the culture collection of cryophilic algae. These were all really worthwhile exchanges and interactions, and what I'm wanting to kind of talk about today is one of the topics of discussion that arose through these interactions, and something that I'm particularly interested in, and that's kind of approaches to understanding community composition in what are very mixed assemblages and mixed communities of algae in these bloom occurrences. So the talk today is going to be relatively general. It's going to be an introduction to snow and ice algae. I know that this is MASTS and it's the marine science kind of seminar series. So this is a slightly terrestrial lean on that. So I'll try and introduce some of the biology of these organisms, and then go into some of the approaches that I've been exploring and some of the techniques that we're hoping to put in place to understand these communities. So starting off, what are snow and ice algae? They're a really interesting group of what you might call an extremophile organism. They're photosynthetic microalgae, and they're able to not just survive, but also to thrive, to grow actively in really extreme conditions on snow and ice surfaces. So they're what you might call a psychrophile. So this is something that has a preferential kind of lifestyle at lower temperatures rather than higher temperatures. So many of these species grow more effectively and faster at temperatures that are near zero than temperatures that are higher than that. They're found all over the world, but particularly in polar high latitude regions and also alpine regions. So areas such as the Arctic, the Antarctic, but also the Alps and the Rockies and the Andes. All of these environments are very much characterized by real extremes in kind of physical and chemical conditions. So they're regularly exposed to extremes in temperature, freeze-thaw cycles, for instance, extreme levels of light radiation, both in terms of prolonged darkness in polar night, but also in high levels of UV during summer months, and also from the kind of reflectance and scatter of light in the snowpack itself. And there's various other stress factors that they're kind of exposed to on a regular basis. And so many of these groups have responded to that by developing these quite incredible kind of protective mechanisms. Some of them might be, for instance, the pigmentation that you can see, hopefully, in the images on the right-hand side. So some of these pigments are kind of photoprotective. They offer them a degree of protection from high levels of UV. There's a number of other metabolites and compounds that they're able to produce to help them in this environment. Things like ice-binding proteins and extracellular polysaccharides, which gives them a degree of physical protection from freeze-thaw cycles and other stresses. Some of the groups are able to kind of swim through the snow, so they have a mobile flagella, which allows them to migrate away and from stress, for instance, from high radiation, high solar radiation during the day. And in many cases, they also have various mechanisms to essentially go into a dormant encysting phase during periods of the year where conditions aren't ideal for growth. So they're a really interesting group, as many extremophile groups are. But beyond just being a kind of curiosity, they're also quite important ecologically and in terms of various kind of biogeochemical cycles. And also in terms of their biotechnological applications and opportunities. So there's various compounds that they can produce which have been used in a whole suite of applications, including Mibel Biochemistry Group here's skin creams, but also in, for instance, their use as a potential candidate for space exploration. They're a very hardy group of organisms. In many polar and alpine environments, they're a very important primary producer. If you imagine, many of these environments have relatively low numbers of primary producers or photosynthetic organisms, and those that are present there are often photosynthesizing relatively kind of slowly. In contrast, the snow and ice algae that we've observed in Antarctica cover relatively large extents of snow and ice, and are able to bloom very rapidly. So they're producing a large amount of organic matter in these environments. That then feeds into a kind of a relatively small but a very specialized and niche trophic or food web in itself. So there's various organisms which then feed on the snow algae, both at a kind of single-celled protist level, but also small invertebrates such as the insect or the springtail that you can see in the top right picture there. Now, more recently, there's been a growing interest in one particular feature of snow and ice algae, and that's that they're essentially habitat engineers, what you might call a cryo-engineer. So a habitat engineer is able to influence the habitat around it to its benefit. And in the case of snow algae, what you have is this very dark pigmentation. So the photo here shows a mixture of the red pigmented sanguina cells, and then these dark black sausages, which are an ice algae and Solanema. The pigment allows a degree of protection from UV exposure, from that stress. What it also can allow is the absorbance of some of that UV energy and re-emittance as heat. This then creates a kind of small localized melting effect on snow and ice crystals around the cell. And for the algae, this is a huge benefit as it provides liquid meltwater that can be used for various sort of metabolomic or metabolic processes, including photosynthesis. So this is really good for the algae, good news for them, but perhaps not so good in a warming world for various ice caps and glacial systems. And there's a lot of work being done now to understand what the additional contribution of this biological factor is on glacial melt dynamics. So this paper by Cook et al. Kind of highlights just how significant that is. They found that in localized spots on the ice cap, on the Greenland ice cap, the accumulation of these ice algae, this biological material, was contributing to an additional acceleration of up to 26% of glacial surface meltwater. So they're relatively significant in terms of these large-scale biogeochemical cycles. And I don't have to point out that if you have accelerated melt, you've then got effects on, for instance, sea level rises, loss of freshwater systems in terms of ice cap melting, and a whole host of effects on albedo. And so there is a growing interest in trying to kind of build effective models of algae growth in snow and ice systems. So understanding growth dynamics, understanding distribution and occurrence, and how they respond to environmental changes, and in particular in climate change. And then what that impact is on wider biological and kind of landscape level systems. And there's a couple of papers here. The Halbach paper is a particularly good kind of overview of where that research is going. Now, when we talk about snow and ice algae, we're not talking about kind of one or two species. They're a relatively diverse group. Relative, some people working in other marine phytoplankton groups, for instance, might not agree, but the images, the panels on the right here, show a selection of species or cell types that were dominant in their particular bloom during a field season that we undertook this year in Antarctica. And what you can see from that is that there is a diversity, not just of species, but also of kind of shapes and sizes and colors, pigmentations as well. And so that represents a kind of taxonomic diversity, but also a functional diversity. So a diversity in how they interact with the environment around them. Now, as well as that, they're also fairly mixed in many cases. So a bloom will not necessarily be a unialgal bloom. It's not just one species. It's potentially a whole assemblage of different species all blooming at once. So the point I was trying to make was essentially that these quite mixed assemblages and this diversity of species and functional groups makes the kind of inclusion of a biological factor in a lot of these models quite complicated and quite difficult. And if you kind of reduce that biological factor to something very simplified, then it loses some of the nuance that exists between different species groups that form these blooms. And so by understanding kind of the community makeup and the identity of the organisms which are included in this, you gain a lot more ecological and sort of physiological insight into the community that you're trying to, or the biological factor that you're trying to model. And then ideally then that leads to improved modeling of that in the wider kind of purpose of that model. And so that's the justification for trying to really understand these communities a little bit better. And so what I've been trying to do over the last year is find ways to kind of capture as much of a picture of community composition as possible. What we're trying to kind of gather when we're doing that are various levels of information. Firstly, the kind of morphology of the community. So what do the cells look like? This acts both as an identification tool, but also in terms of understanding the kind of ecological traits of those groups as well. Really important to gain some observational data of occurrence on a landscape level and the environment in which these blooms are occurring. And then beyond that, what's very useful to have is some idea of the phylogenetic identity of the organism. So where does it sit in an evolutionary context and relative to other organisms that are perhaps better understood? If you're able to place it in terms of its phylogeny, you're perhaps able to gather some more insight into the ecology or the kind of ecophysiology of the organism from related species. And then finally, really important is having that kind of overall community analysis. So that biodiversity level of data, looking at things like the richness of the community, species richness, the evenness of species within the community, and which species are dominant and which species are perhaps rarer. And in some cases, associations between key species too. Now, the tricky thing is with snow and ice algae in particular, it's never as straightforward as it seems. These organisms clearly grow in very difficult places to sample, often quite high up mountains or in the poles. So it makes sampling quite tricky. The habitat and the blooms themselves are fairly ephemeral, so they're never always happening where you might expect them to. Difficult to predict how long a snow patch might last, and they're affected by all sorts of things like rain washout. An additional complication is that many of the species are very difficult to tell apart by morphology alone, so just by looking down a microscope. They're what you would call a cryptic species. So it's only when you look at evolutionary kind of relationships, the phylogenetic placement, that it's easier to tell apart whether two cells that look similar are different species or not. And then conversely, there are a number of species which have various life stages that look very different under the microscope, but perhaps are one species. And with many of these organisms, the life stage or the life cycles are still very much an unknown factor. And so taking that into account can be very difficult as well. And so what I've done here is try and just outline what the various kind of approaches are that exist for assessing communities, what the kind of advantages and disadvantages are. And I won't go into them in too much detail because I realize I've now wasted time with my video. But what you have is essentially these kind of macro level observations. So observations at a landscape level, what size of the bloom is, where it's occurring. And one of the benefits there is you can use things like remote sensing, drones and satellite data to improve that. The next thing to do is to get the sample under a microscope and assess it visually. So that gives you this morphological ID. So you're able to at least place where it is compared to other cells that you might have seen under the microscope before. And also gives you this trait kind of level of data. So cell size, shape, color, and for instance, how that might affect its ecology. And with microscopy, you can also do kind of qualitative and semi-quantitative assessments of community as well. So either using kind of SAC4 style abundance metrics or using kind of more quantitative cell counts. But really, what you're needing for phylogenetic placement for kind of a more precise identification of the community members is some molecular data. And there's two approaches to this. Firstly is sequencing individual kind of cell types. Traditionally, this was done by isolating the cell, growing it up into a bulk biomass, and then running an extraction PCR and sequence from that. But increasingly, people are looking at how we can essentially pick apart individual cells and do what's called kind of single cell barcoding. When you combine that with an image, what you have then is a catalog that slowly builds up of these cell images combined with a sequence, which gives you the phylogenetic placement or the phylogenetic identity. So they're quite powerful approaches, but still quite laborious and still require some tweaking. And they're also quite affected by kind of human selection biases. And finally, what's used increasingly is metabarcoding or eDNA approaches. This is where you sequence a whole mixed kind of sample from the environment, and it'll give you a whole list of barcode sequences back that give you an idea of where all the different organisms sit in your phylogeny and your evolutionary tree. This is really beneficial because it's a kind of whole community approach, or at least it gives you a slice of the whole community. What it also gives you is a semi-quantitative assessment of biodiversity. So you've got things like species richness, species evenness, which can be really useful, in particular in combination with microscopy. And this kind of gives you the pluses and minuses of each of them in relation to those target pieces of information that we want for the community analysis. But the takeaway from this is essentially that you're better off not relying on one approach. You're better off trying to combine a couple to get the most out of the analysis. And so this is what I've been trying to do in the last year, is set up a kind of a pipeline where we use a number of the approaches and then combine the outcomes of those approaches to kind of improve or enhance on the individual outcomes. So you end up with these kind of synergies between the different data sets, which actually give you a greater product than the data sets individually. And so this was trying to summarize that, although I realize the arrows are a little bit messy. I need to probably remake that figure at some point. This summarizes a few of the main benefits of combining these approaches. One of the first ones is that by having this single cell sequencing and image catalog in combination with a metabarcode, you're able to do all sorts of kind of improvements in terms of improving the phylogenetic resolution of your metabarcode data set. If you have your metabarcode read, you're able to align that against your catalog of images and sequences from the single cell barcode reads. You're then able to essentially give a lot more phylogenetic resolution to key reads in your metabarcode data set. And you're also able to include a kind of ecological trait, morphological data from the microscopy to the metabarcode data set. One of the downsides of metabarcoding in many cases is there isn't a kind of a direct link between the reads in your metabarcode data and the microscopy that you're seeing, or the cells that you're seeing down the microscope. And this kind of bridges that gap to give much more ecological context to the metabarcode data set. The microscopy also acts as a really useful kind of qualitative validation of the eDNA or metabarcode data set. So you're able to kind of get an assessment of the community through the microscope. And if that looks drastically different to your metabarcode data, then it's probably worth having a double check whether you're getting some sort of bias occurring at one point or another. So they combine very nicely and complement each other. And then finally, one of the problems with the microscopy approaches is that a lot of these cryptic species and life stages are very difficult to detect. And again, having metabarcode data and the single cell data then fills in those gaps. So the metabarcode data can give you an indication of whether there is a large amount of hidden cryptic diversity within your community. And the single cell sequencing can give you an idea of whether different morphologies of cells that you're seeing are actually different life stages of one species. So all sorts of really interesting synergies and kind of benefits from combining these approaches. And so this is kind of the final outcome. You have this qualitative and quantitative community analysis from the combination. Ideally, you have a high resolution phylogenetic placement of all of the key members of your community. So you're able to identify them at a species level if possible. You also have the morphology of the key community members. So you're able to do things like trait analysis across an ecological kind of gradient or survey. And you have the detection or a high level of detection of these cryptic species and life stages. And so one of the aims from the Antarctic work and some of the collaborations with Norway has been to build a kind of cell image sequence catalog. And once that sequence catalog is built up, ideally, you then don't have to continue with that single cell work, and you can move to assessment by purely microscopy and metabarcoding alone and link it back to the existing catalog that you have. So I think I'll probably skip this because I don't know what the time. Oops, I don't know what the time is. And I'll finish on that note and just say thanks to everyone who was involved at all parts of the project. It was a really, really enjoyable and worthwhile program. And yeah, a special thanks to Thomas and Chris and Alex and Hirono, who were really wonderful to kind of have these discussions with, and hopefully we'll continue working together. Thanks.
Thank you very much, Alex. It was a shame about your video, but I got to see it. It's very cool. So now we'll move on to Ximena's talk. If you would mind sharing your screen, please.
Okay, well, thank you very much, Alex, for this great talk. It was super interesting, so thank you for that. First of all, I would like to thank, of course, all of you that are now watching this webinar. But I specifically would like to thank Saltire for for supporting this project that actually has been getting bigger and bigger. But of course, for that we need a first support, so then we can start or continue building up on this kind of projects and mostly as an early career scientist. So I'm super excited to share with you some of the advances of this project titled Organic Carbon in Polar and Subpolar Glacial Marine Systems. And well, thanks again for everyone for watching. Okay, so let's start with glaciers. We already knew a lot about glaciers. I mean, we knew about their dynamics, their physical characteristics of glaciers. But just about 30 years ago, we found out that actually we didn't know very much about glaciers, because the thing is that, as Alex mentioned, some areas of these areas covered by ice or snow actually are quite productive, which means that these areas have a very important role in the carbon budget on the planet. So let's move a little bit deeper into the subjects of my research, which are glacial marine systems. So what is a glacial marine system? Well, as the word says, a glacial marine system is this system composed by a glacier and the marine system. Now, why are these glacial marine systems so important? Well, the thing is that they are super powerful CO2 sinks. And it's quite fascinating as well to see how these pieces of frozen ice communicate to these pieces of liquid saline water. So looking at these super complex processes, it's what I'm looking at, but specifically everything that has to do with organic carbon that is being produced on the surface of glaciers. Well, not just on the surface, but also within and on the base of glaciers. So I would like to highlight this number 1.5, and I'm pretty sure that all of you that are behind this screen watching this webinar know exactly what I'm meaning with this number 1.5. We all know that this is a very famous number, and we should, keep this number as far as we are able to. But the bad news is that for the ice in our planet, this 1.5 is not even enough, because most of our glaciers have already absorbed so much heat that they will continue to melt during the next years, including far from, well, by the end of the century. So why I decided to mention this? I decided to mention this because we as scientists, or the people that are looking or studying polar regions, have to be, well, we have to make sure that we understand the actual processes to feed on models and feed on the projects that are looking at the probable outcomes of this context of climate change within these super important regions, which are, it is important to mention, the most vulnerable systems right now in this context of global warming. So my project, the aim of my project is, or was, to characterize the properties of carbon-related constituents and the underwater-like climate in 6 glacial marine systems as a first step to describe regional differences. Why I wanted to look at regional differences with regards to how glaciers deliver organic carbon to the marine systems. Well, the thing is that although we might think that glaciers, well, actually glaciers are in general a quite simple element on the cryosphere, because they grow when the temperature is low, and they get smaller when the temperatures rise, or they get smaller because of some inherent dynamics of the glaciers. But they also respond to microclimates. So this means that regionally, glaciers respond very, very different from one place to another. So I wanted to make sure to have not a very clear picture of what is happening in each region, but to try to understand the processes with regards to the geography of these places. So the parameters that I decided to look at is color dissolved organic matter, which is the colored fraction of organic matter. And this is quite important, because this fraction of organic matter allows us to look at, for example, the concentration or the distribution of organic matter from products such as satellite images. Now, fluorescence, because if we measure fluorescence, we can get some ideas about the chemistry or the origin of this organic carbon. Of course, I decided to take measurements for concentration of dissolved organic carbon, particulate organic carbon, but also particle absorption. And this, why this is important? Well, this is important because with these amazing devices, such as the one that you can see on your right, on the right side of your screen, we can measure, or we can know exactly what type of particles are within the water. So let's say we could find in these marine systems inorganic or organic particles that can be related to the organic components or organic related particles in the marine system. I also decided to take into account the bioptics to characterize the optical properties of the marine system, because although we have these amazing tools of Earth observation, such as satellite images, we don't have very good products yet, mostly in polar regions and subpolar regions. I'm sure that the people that have or is working in these areas understand exactly why. So the idea was to go to these places, characterize the underwater light climate of these systems to validate, but also to improve the products that are already available for all of us. So what you're looking at, these are photos from the interchange that I did at the University of Bergen, and this was because of the Saltire program. So thank you very much for that, because I learned a lot, but I also met amazing people. And this visit to the University of Bergen allowed me to have a new network, but also to expand a little bit my project. So as I mentioned, I wanted to look at the differences for each region. So I have had the opportunity to participate in a couple of Antarctic campaigns, such as here in King George Island in 2019. I also had the opportunity to be part of an expedition in northern Patagonia, and with the University of Bergen, within the context of the Saltire interchange, I got the chance to go to Lurefjorden. And because of this visit to the University of Bergen, I also had a chance to get this amazing data set for Gavnefjorden. So this means that I've been building up a huge amount of data, which will allow me to look at these regional differences that I would like to see with regards to the input of organic carbon to the marine system. Now, it is important to highlight that these are just first results. To be honest, the first 2 years of my PhD have been mostly focused on fieldwork. So I've spent a lot of time outside in the field, on the ice, on the snow, and on the ocean. So I haven't had much time to look at the data or to process the data. So it is important to highlight that this is just just a taste of the results that I have been able to process so far. What you're looking on your screen are plots of serum absorption spectra. And what this means is that depending on the value of the curve is the concentration of this colored organic carbon on the marine system. So for instance, you can see that most of the curves that you can see on the right side of your screen are below 1.5. This means that most of them have, in average, the same concentration of this colored organic carbon. But for example, you can see that this orange color, which is represented by the northern Patagonia, is very high with versus the other areas. And this is quite interesting because in northern Patagonia, we have a huge presence of marine terminating glaciers, but not just that, but also a lot of vegetation and many possible sources of organic carbon. Now, what it is important to highlight is that I'm not just interested on the concentration of organic carbon, but also on the characteristics of this organic carbon. Because depending on the characteristic of this organic carbon will define the impacts of this organic carbon on the overall system, on the overall marine system. So it is important to highlight these differences in concentration, but I will look deeper into the chemical characteristic to try to understand what might be the impacts in the near future. So what you're looking at on your screen now are the plots of concentrations of particulate organic carbon and dissolved organic carbon. I will just highlight the values, for example, of Yelcho. This orange figure is represented by a Chilean research station, which is called Yelcho, that is at 64 degrees south. And as you can see, in average, Yelcho showed the highest concentration for both POC and DOC. And King George Island, which is this small island in the Shetland Islands in Antarctica, showed in average the lowest concentration of DOC and POC. And Lurefjorden, which is the fjord that I visited with the people from the University of Bergen, showed high values, the highest values of POC, whereas for DOC, the values were the lowest. And for Southern Patagonia, we didn't have the chance to get samples for DOC, but well, the amount of POC was a bit higher, well, very high versus King George Island. But as you can see, this can tell us a lot about how each region is responding to these processes of melting, and not just the inputs of glaciers, of course, but from other sources that might have a big importance with regards to the presence of organic carbon. And because of that, I wanted to see or have a quick look into the relationship between these colored organic carbon and these other organic components such as POC and DOC. And as I told you already, just to highlight and just to remember that these are just initial results, but I found out that, well, a negative correlation between POC and DOC, and a positive relationship between colored dissolved organic matter and DOC. Now, as we already know, these complex systems need more than just specific studies, right? We need to understand the processes in a broader picture. I mean, with regards to time and space. So fortunately, we have these amazing tools of Earth observation, these satellite products, but the thing is that we still have to improve them. And mostly, as I've already mentioned, in polar regions. But I tested some approaches, such as the optical water types, to see if we could get some insights on the distribution of some organic components within the marine system. Just to mention very, very quickly, these optical water types were based on this approach developed by my supervisor, Evangelos Pirakos. He took reflectance measurements from multiple sources, both for inland and marine environments. And we used this approach to look at the distribution of organic components in the marine system. And what you're looking right now, it's a Google map from Spitsbergen in Svalbard, and we apply this optical water type by Spirakos, and we found, for example, these green colors we found turbid with high organic content waters. And on the other hand, we also found here in these enclosed fjords, we found a serum with cyanobacteria and high absorption efficiency by NAP. NAP is non-algal particles. So these maps, as you can see, can tell us a lot about what's happening in these highly complex systems, and probably what might be the role of, for instance, some small glaciers or bigger glaciers or just land. How might they impact with regards to, I mean, biochemistry, but also primary production. So once again, we apply the same approach in the east coast of Svalbard, and this time we found higher serum-rich waters and hyperproductive cyanobacteria abundance and sediments exactly in the coastal areas. But this is not enough. We need to look a bit deeper into some details of the not just particles, but also components that we might find in these systems. And why this is important? It is important because, as I already told you, the chemistry of these organic components will define the impacts that might trigger in the marine system. So I wanted to try a tool with a higher resolution to try to find some spectra associated with specific chemistry of the organic carbon. So for that, I tried with Prisma images. These Prisma images are hyperspectral. So you can get images such as this one. So you can see both the characteristics for both the marine system, but also in land. So what I did is I split it, the land, and I classified some areas with a specific spectra. The spectra means the curves, specific curves depending on the composition of these materials that you can find on land. And as you can see, we found different characteristics, which means different particles and different chemistry that end up in the marine system. And this is the marine system. I split it, as I mentioned, land and ocean. And we found these patches of different components, as we did with the optical water type approach. The thing is that, of course, this is a relatively new product, Prisma, these hyperspectral images. And for that, we still need, it is super necessary to go to the field to characterize all these waters and also land, both snow, ice, and ground, to get all these specific spectra to validate and improve these fantastic products that we have available for us. So what is next? As I said, I identified that we have still a huge gap with regards to the optical characteristics of organic carbon-related particles or components, both in land and the marine system, that is in the glacial marine system. So for that, we need more data. And thanks to this first seed, which was the collaboration between the University of Bergen and the University of Stirling within the context of the Saltire program, I had the chance this year to travel to both poles. In January and February, I was in Antarctica taking samples for all the parameters that I've already showed you. And in April, I traveled to Kongsfjorden in Svalbard in the high Arctic to do exactly the same, to do all these measurements and characterization of the bioptical properties of both the ocean, snow, and ice. So here am I taking some measurements for snow. I took samples, but also radiance measurements, and I did exactly the same in the marine system. I took water samples, and I also deployed devices to measure absorbance and backscattering. And this is me again in the Arctic. On the right, you can see me holding an ACD to measure reflectance on sea ice. And on your left, this is an optics box. That's how we call it at the University of Stirling. And with this, we can measure all the optical characteristics in the marine system. So what am I doing now? I'm awaiting for exciting results to have more information about the chemistry, the concentration, but also the optical characteristics of all, or as much as the optical characteristics of organic carbon as I'm able to get. So I would like to thank once again the Saltire for supporting my idea, my project, for believing in me, and of course all the people that has trusted in my project and that has been helping me both in the field but also in some administrative stuff. So thank you very much to all.
Thank you, Ximena. That was really interesting. And it was just interesting to see the different perspectives on polar regions from you both, biological and physical. So thank you very much. Just a reminder to everyone watching, you can put a question into the Q&A box and we can get to it, either for Alex or Ximena. We can ask them both questions. So I actually have a question for Alex first, if that's okay. Are you there, Alex?
Yeah, just here.
Yeah. You worked in Antarctica, was that right, for your samples?
Yep. So that's what the group concentrates on. So we were down for a couple of months this winter.
So do you just find these species down in Antarctica or maybe in the Arctic, or can you find them in different places, perhaps the UK?
Yeah, exactly that. You can find them all over the place. So they've kind of been able to colonize anywhere where there is snow and ice and a degree of kind of melting and meltwater available. So we've mainly looked in the Antarctic, but increasingly I've been quite interested in looking more locally and trying to see if we can document them in Scotland, which is a very different environment. Snow doesn't last nearly as long, so there's only a very few number of locations where snowpacks survive into the summer, and sadly those are going to likely disappear in the next few years as temperatures warm up. Yeah, last summer we made well, it wasn't the first discovery. There's a documented snow algae from around the 1960s, but not very much since then. But yeah, we've kind of made a bit of a rediscovery of a couple of patches in the Cairngorms and around Ben Nevis, which was really good fun. Yeah, hopefully we'll be able to do a bit more kind of analysis and bring those to light a little bit as well. It's kind of a hidden biodiversity of the Scottish Highlands that hasn't really been looked at. And actually that was in collaboration with Alex Dittin, who was the PhD student who came across from Nord University to Scotland. So yeah, that was again funded by the MASTS-Saltire exchange. So that was really a nice additional kind of output from that.
That's great to hear. We actually have our first question in the box for Alex. So Alex, to which degree do rare algal species occur in metagenome analyses?
Yeah, I think tricky one because it depends a little bit on the barcode that you're looking at and the thresholds for rare species that you're comfortable with. So it becomes a little bit of a statistical question as well as an ecological question. Yeah, so I'll be getting into that a little bit later on. Yeah, I don't have a definitive answer for that right now. I think, again, one of the benefits of having, for instance, the microscopy is that you can kind of just validate some of these rarer species, or at least have a quick sanity check. You know, if you're getting these species that are occurring in the metabarcode data set, hopefully you're then able to align them to a phylogenetic tree, and you're able to get an idea of where they sit and what kind of group of organisms they are. And then in an ideal world, you might then observe what you think is the organism under the microscope as well. So there's ways of improving the support for some of these rare occurrences in metagenome analysis. But yeah, the kind of the hard threshold, the cutoff is varies depending on the parameters of the data set. Yeah, and whether they could be overlooked in metagenome data is another really important point, and I think it's often one of the kind of biases that should be taken into account. As sequence data becomes, well, as some of the sequence technologies and algorithms become more specific, then accuracy improves, and you're able to say with more confidence that rare sequences are supportable. So that helps as well. But yeah, certainly a bias to take into account.
Thank you, Thomas, for that question. Ximena, I actually have another question for you this time, if that's okay.
Yeah, sure.
So were there any surprises for you? Were you expecting something, I don't know, thinking about what glaciers release versus rivers, or what's in lakes, other bodies of water, that sort of thing?
Yeah, that's a very good question, because some people have asked me whether, I mean, you see glaciers and they just look white, right? And you expect that they are just, I mean, completely rid of life. And the thing is, of course, glaciers do have, lower concentration of organic matter, but the question would be, what type of organic matter does glaciers deliver, right? Because we have seen that the organic matter that comes both from rivers or land is quite different. And I mean different, for example, the organic carbon that comes from, for example, the microalgae that Alex studies is quite fresh, which means that it's quite labile. And if it is quite labile, it can be metabolized much faster in the marine system. So perhaps the impacts that might have the smaller amounts of organic carbon coming from glaciers to the marine system might have a stronger impact than bigger loads of riverine inputs to the marine system. So yeah, that's super interesting.
Thank you so much. So I don't think we have any other questions coming in right now. Oh, I spoke too soon. There's one again from Thomas. So Ximena, does your method of hyperspectral look through snow? Regarding Svalbard in May, June, there is still a thick snow cover. So how can you look at the soil/substrate? I suggest you go there in July for a field trip.
Yeah, well, thanks for the question. I'm not interested on soil. I'm interested on snow, because although you just see it white, it still has organic carbon. So the idea was to characterize the optical properties of snow and to take samples of fresh snow to see the amount of organic carbon on snow. Although it would be quite interesting to see both, like going to Svalbard in winter and late in July, perhaps, to see some differences on the types of carbon. But thank you so much for the suggestion.
Thomas follows up saying, in June you can look at all the different sediments from all Earth times. Maybe the underlying solid affects the snow.
Yeah, of course. Yes, that's right. Exactly.
So I think we're just a little bit over on time. So, oh, do we have time for one more, Ximena? Is that okay? There's one for you.
Yeah.
From Pamela, she says, do you have an idea why the amount of carbon is much higher in Patagonia?
Well, we think that, or my guess is that, for example, in northern Patagonia, we have lots of marine terminating glaciers. So all of them are melting. So probably that's one reason, that perhaps the amount of organic carbon that is coming from glaciers, because they are melting, is higher. But also the presence of vegetation, for instance, in northern Patagonia is quite important. If we compare it with, for instance, with Antarctica, with King George Island, which is very close to Patagonia, but still the microclimate and the geography and the geology, it's different. So that's why it's super interesting to look at different regions, to look at how each actually glaciers, but also the inputs and the characteristics of organic carbon might drastically change, actually, from northern to southern Patagonia, actually.
Thank you very much, Pamela, for that question, and for Ximena for answering it. I think now we're done. I've said it a few times now, but I think that might have been the final question. So thank you everyone for listening today, and thank you to our presenters. Just before you all shoot off, I would like to mention we will have another talk in a couple of weeks from Emma O'Halloran, and that'll be on sea urchin parasites. Quite a lot of diversity we have between webinars. So information and details on how to sign up will be on the MAST website, and we'll also circulate it via the mailing list and the Twitter account as well. So yeah, thank you again, and hope to see you there.
Thank you so much.
Thanks.
Bye-bye.
Bye.
Bye.